Hi, good morning, everybody. In the last class, we were looking at this idea that hippocampus plays a role as an important structure in the memory functions of the brain, and we discussed that it's like a scratch pad of memory. Memory stays in hippocampus for a short while before it is shipped off to long-term storage in the cortex. So this is a very big, system and very complex system, and we don't know all the pieces of the puzzle. But in this class, I'll only be focusing on what hippocampus does and how things are stored in hippocampus. Even that according to certain theory. I'm not saying that the theory is perfect or completely right or anything like that. I'll just talk about one of the major theories, and but I'm not going to talk about how information goes from hippocampus to the cortex again. Okay, so yesterday this is where we stopped. We looked at the anatomical relationship between cortex and hippocampus. We have bidirectional connection in cortex and hippocampus. It happens through many stages. It's not direct connections. And so I just gave an intuitive description that so if you have a cortical state, consisting of multiple sensory modalities, like color and hardness and just like related to touch. All of them project to this via with multiple pathways to a small system which is hippocampus. So there's a dimensionality reduction from the high dimensional cortical state to a low dimensional hippocampal state. So in that process, it generates a compact representation of this high dimensional state. And now by the feedback connections, that low dimensional state, which is in the compressed form of the cortical state, is reconstructed back in the cortex. So this is a kind of picture that we described yesterday. So in this context, I want to mention the idea of bidirectional associative memory. Because hippocampus is a Hopfield network is an associative memory. We have seen that. And in a Hopfield network, you just have a bunch of neurons where everything is connected to everything else. Right, so it's a fully connected system. But just imagine a Hopfield network in which I just split the network into two halves. The total is n, whereas here I say this has p neurons and this has q neurons. So then I can think of this as two layers. One has p, another has q, which can have different sizes to be general, right? And you have connections going from this system to here and this system back to here. And of course, there are connections within the layers also. Okay, so something like that. Now, how will this system work? So normally in a hippocampus, you start from V0, right? Then the update will takes you to V1 and so on and so forth, and it stabilizes. When you have a system like this, where you just notionally imagine that the hippocampus, the Hopfield network is divided into two sub-networks, one has P and one has Q. Then you give the input to this, I'll call this X, and output of this is Y. So I start with some initial input, X naught, that gives me initial output, Y naught. And Y naught is fed back, right, to X, and that gives me the next state of X, which is X1. And X1 is fed forward into this network, and that gives me Y1, and so on and so forth. The thing is, the same thing as what happened before is again happening here. Only thing is, I've split the network into the network state into two halves, two parts, not halves, two parts. And so the update is happening alternately. Now we know that the original Hopfield network, in the original Hopfield network, update can happen in online mode where single neuron is updated at a time, or in batch mode where the entire state is updated at a time. So think of this as some kind of intermediate mode where a part of the network state, which is defined as X, because the whole state V is nothing but X concatenated with Y. Okay, so you just imagine a kind of intermediate update tool where X, this whole thing is updated first and then Y and so on and so forth. Okay, so that this gives you X2, and now you can easily show that the same energy function will be valid even for this kind of an update. The update doesn't matter, right? We know that energy function simply determines the minima irrespective of your pattern of update. So this also will go to a stable state. In the final stable state, for a given x, you will go to you will go to a given y. Okay. So, and what is nice about this kind of a system is, unlike a perceptron. So if you look at a perceptron or even an MLP, right? For a given input, you get an output x, and there ends the matter. There's only, it's a purely feed-forward network, and there's no feedback. Whereas here, there's feed-forward and feedback, and for a given x, you get y, and alternately, you can also start this network in the different direction. I can start with y0. I can give y0 as input to the system, initiate the process, and y0 then give me x0. Then x0 will again give me y1, and so on and so forth. So both are identical. I mean, both, there is no preferentiality over x of x over y or anything like that. Now this is a bidirectional network. A lot of nice things can come out of this, because imagine that, let's say, x is a small layer, okay, and y is a much bigger layer, and so you can have a compressed representation of y in the form of x, and so that's kind of like what Hippocampus is doing. And not only that, you can think of there are other generalizations of this, where you can think of this as split into many layers, right? Where the connections are in a feed forward and feedback direction. Now that brings us even closer to what is happening between cortex and hippocampus, because in the cortico-hippocampal connections, we have seen that we start from the highest layers of cortex. So this mainly the sensory association cortex and the prefrontal. Okay, slightly higher order cortical areas, not the primary sensory cortices, but slightly higher order cortical areas. Project to parahippocampal and perirenal cortex, and then to entorhinal. So it's happening in 2 stages. So that kind of thing you can easily realize as a generalization of the Hopfield network. Okay, and so that's the kind of memory you can visualize, right? That hippocampus is. Okay, so so far we only talked about the connections between cortex and hippocampus, but we didn't say anything about what is happening inside the hippocampus or the hippocampal complex. Now, the entry, so the cortex projects to this multimodal associative cortices, project to EC, the entorhinal cortex. And this is also a cortical area, so therefore there are multiple layers in it. This is layer 3, layer 3 again, and then layers 4 and 4 to 6, and so on and so forth. So from here, the information goes into the hippocampus. The hippocampus consists of multiple neuronal clusters. So the input, and actually it goes through a loop of connections, right? Input into the hippocampus goes from the more superficial layers of, so this is layer 2 and this is layer 3, more superficial layers, and it goes first to a place called dentate gyrus, DG. This connection between EC to DG is called a perforant pathway. Then from DG, you have another set of connections going to another cluster called CA3. And within CA3, there's lots of recurrent connection. That's a very big, interesting feature of CA3 connections. Then CA3 projects to another cluster called CA1 through a set of fibers called Schaefer collaterals. Okay, so then EC also directly projects to CA1. So this is called temporoammonis pathway. And CA3 projects to subiculum. Subiculum projects back to the deeper layers of EC, and deeper layers project back out to the cortical layers. So, okay, we were thinking of a kind of a bidirectional associative memory between cortex and EC. But then EC does a lot of stuff inside the hippocampus, and that is a whole different story. So that's what we'll discuss now in this class. The other minor connections, for example, the EC, the superficial layers of EC also project directly to CA3. They project to DG through slightly stronger connections, but through weaker connections, see this dashed dotted line, this kind of faint dotted line. It also projects to CA3. And so there are multiple connections. Basically, this is typically anywhere in the brain. This is a standard feature. But I'm only talking about the main connections. So this is just a description of what I just said with that using that picture. Uh, so also a lot of these connections, the mossy fibers and Schaefer collaterals, these were subjects of a lot of LTP studies. Because hippocampal connections are highly plastic. Plastic, plasticity means changeability of synapses. That's called plasticity. Okay, so hippocampal synapses are very plastic, and these mossy fibers, they were all, they've been studied extensively by the LTP community. Now, where does Hopfield network come into picture here? Because we talked about relevance of the Hopfield network type of ideas when it comes to the cortico-hippocampal connectivity. But you can apply these same ideas in a different way in one of these deeper hippocampal systems. In fact, that is the main story. So there's a major idea or a major theory, which is what I was talking about all along, that CA3 is some kind of a recurrent network, something like a Hopfield network. So why is that? Because CA3 is special in the sense CA3 neurons have extensive recurrent connections. That means you have the feed forward connections one layer to another and have recurrent connections. Thing is, you have recurrent connections everywhere in the brain, but the CA3 connections are supposed to be slightly more recurrent than other areas of the brain. So, for example, in the rat, there are about 300,000 pyramidal neurons. Pyramidal is a very important kind of uh, uh, neuronal type in the brain, found extensively in the cortex, also found in some subcortical areas like CA3, for example. So with each of them receiving inputs from about 12,000 other CA3 pyramidal neurons. So Hopfield network, for example, is an example of total recurrence. Every neuron receives inputs from every other neuron. So if you have n neuron network, each neuron receives n inputs or n minus 1 inputs. Whereas here you have 300,000 pyramidal neurons, and each neuron receives inputs from only 12,000 other pyramidal neurons. Okay, but even that is considered to be high, very high in the brain, and this is in fact the anatomical studies state that this is highest in the brain. So whereas in contrast, each pyramidal neuron receives only 4,000 inputs from the EC. So they have much higher recurrence among themselves compared to the number of fibers they get from outside. So this is an anatomical feature, and it's very hard to argue against that because anatomy is kind of sacrosanct. Each CA3 receives therefore and from inputs from about 4,000 other CA3 pyramidal neurons. This is high recurrence and highest compared to any other brain region. Another interesting thing is these recurrent connections are modifiable by LTP, so they are plastic. And so these anatomical features, these special anatomical features of CA3 have led people to imagine that they act like some kind of a Hopfield network in which patterns are stored. But then when you want to store patterns in a system like that, we have gone through all these arguments from a mathematical point of view. That is obviously if you want to use it like a memory, it has to have two modes. It must be a storage mode and retrieval mode. And anatomically, it should have high recurrence, and the synapses should be plastic, and so on. So these are all things that you need if you want to develop a theory that CA3 is a storage system. So here I'll use a classroom analogy. We have gone through these arguments yesterday with using the equations as a prop. But now let me just discuss that in more intuitive terms. So imagine a classroom where the teacher is telling something, conveying some knowledge to a bunch of kids. And so when teacher is explaining the concept, obviously the students should be quiet. They shouldn't be chattering among themselves. They should only listen to what the teacher is saying. So the connections from teacher to student must be strong, must be very influential. Connections from among students should be weak while the lesson is going on. Okay, so that you can call that the storage mode. Whereas in retrieval mode, when teacher is asked the class a question and then they're supposed to answer. Now, let us imagine this is a more democratic class and where students can discuss among themselves and come up with an answer. Because let's say it's not a simple kind of a factual type question, but more thought type question where you need some to think deeper into the question. So when in this mode, when the retrieval mode, when teacher is asking question and class has come up with an answer, the inputs from teacher to student should be somewhat weak. Because you just so question is more like a suggestion, like a cue, and so therefore that should be weak. And then the now students get into exaggerate chatter and they discuss exaggerately and all that, and come up with a response. So in this mode of operation, the connections among the students must be strong, right? Whereas the connections from teacher to students must be weak. Okay, so similarly, you would imagine similar things are happening in CA3, or they should be happening if CA3 is operating like some kind of a Hopway network. So which means during storage, external input is strong, recurrent connections are weak. They are weak, but they are allowed to be plastic. They must be highly in a changeable state. Okay, so CA3 strong into external input, recurrence is there, and the connections within CA3 should be plastic. This arrow cutting across the box indicates that the CA3 is in an adaptive state. It is a malleable state. Whereas in retrieval, external input must be weak, recurrence must be strong, because that's how the network retrieves. And recurrent connections must be strong but not variable. We don't want them to learn in that time, right? Because so what is happening when from the state the connections learn, and from connections the state develops. So there is a mutual dependency. So both cannot change simultaneously. One is fixed with respect to that, the other one develops. So during this state, recurrent connections are not plastic, and but they are strong. Now, is all this really happening in CA3, or is it only a mathematician's hallucination? Now, interestingly, we know that CA3 has two external inputs, from EC via perforant pathway, and from EC via dentate gyrus by mossy fibers. So there's a direct connections from EC to CA3 via perforant pathway, and indirect connection from EC to dentate to CA3 via mossy fiber. So we have seen all those connections here. Yeah, so direct connection from EC to CA1 via perforant pathway, indirect from DG indirectly from EC via DG through mossy fiber. So there are two connections. Okay, and perforant pathway inputs are weaker than mossy fibers, and that is one thing. So you can, in principle, control your input strength and all that. But more importantly, the plasticity inside CA3 must be controlled. Now, how is that controlled? So there, people have observed that there is this chemical called a neurochemical called acetylcholine, which has projections to CA3, and which is capable of controlling the strength and plasticity of recurrent connections in CA3. So far we discussed about neurotransmitters like glutamate and GABA and all that, but we never talked about neuromodulators. So these are a different class of neurochemicals which are capable of changing the synaptic plasticity, the strength of a synapse, more rapidly. So normally when the synapse has to change, you have to perform some kind of an LTP. You need to create a LTP kind of arrangement, and then fire the input side, fire the output side, and then do it for a long time, and then the synapse changes slowly. But this is going to take 10-15 minutes. Whereas with the neuromodulator, you just moment the neuromodulator terminal, right? Because these are also chemicals released by neurons. So when a neuromodulating neuron releases neuromodulator onto a synapse, it will immediately change, right? Or order of maybe even milliseconds. So the neuromodulators effect on the synapse makes it plastic. So that's what this picture is indicating. It can rapidly change its synaptic strength. So there are several neuromodulators, important ones in the brain. Acetylcholine is only one of them. The other ones are dopamine, norepinephrine, and serotonin. And interestingly, these neuromodulators are associated with some of the major diseases, brain diseases. You see deficits in acetylcholine in Alzheimer's disease because that's a disease related to memory loss. You see deficits in dopamine in Parkinson's disease, and it's not a motor problem, but again, there's a widespread motor impairment. So in fact, people find that although acetylcholine is mainly affected in Alzheimer's, dopamine in Parkinson's, In fact, in these diseases, you see some kind of deficiency with the function of all the four neuromotor systems, right? In fact, the four work together. It's not that they're all working in isolation. So acetylcholine is important in hippocampus, plays a major role, and also it's impaired in Alzheimer's disease. And blockage of acetylcholine transmission, therefore, you can guess will affect your memory storage, and that has been extensively studied experimentally. So, for example, there's a drug called scopolamine, which is an acetylcholine antagonist, and if you apply that into humans, it will create temporary amnesia. So you might have seen this again in movies. Again, like I was joking yesterday, you learn a lot of things from movies, and so you hear about this brainwashing experiments where you erase memories of somebody and all that. Lot of those drugs act on the acetylcholine system, so that's how you attack memories. So in one such study with scopolamine, the volunteers were given a dose of scopolamine and were asked to memorize a bunch of words. Okay, and so they had little or no memory of the information presented during the test, and some volunteers even forgot about the trial. So it was the memory loss was so bad. That is, some people didn't even remember that they went through this whole experiment, and that's memory loss is so profound. So the controls, right, were able to memorize 45 out of 128 words, but the scopolamine subjects could only remember 6 out of 128 words. And also, if the scopolamine was given after the words were already learned or by hearted, there's no impairment in performance, but only if they were given before the experiment, right, it was able to block the memorization process. Okay, so you see exactly what it is doing in the brain. Now, so that is a very indirect, high level, behavioral level study. But people also have found in simple cell culture studies where you take a slice of the brain and then put electrodes and study LTP in a kind of LTP setup. Also, people have found that acetylcholine controls LTP, and you can modulate LTP by simply applying acetylcholine to your preparation. So Michael Hasselmo, who is at Boston University, and other colleagues have developed these kinds of theories, and have suggested that acetylcholine satisfies the criteria necessary to switch between storage and retrieval. So all the arguments we have presented so far, that any memory system should have two modes, storage and retrieval, and all that, right? And the acetylcholine's role in CA3 still seems to fit the bill very beautifully, because at high ACH level, it's been shown that input is strengthened. So the input to the network, right, to the CA3 system is become stronger. Recurrent connections of CA3 are weakened; they become weaker, and also there's increased plasticity in the recurrent connections of CA3. And at low ACH levels, input is weakened; it's decreased. Recurrent connections of CA3 are strengthened, and there is a decreased plasticity in the recurrent connections of CA3. Okay, so that is also seen. Now, okay, so that is still, but how do you know all this, right? This only almost seems like a hypothesis. How do you know this? So for that, we need to go deeper into the anatomy of CA3. Now, CA3 has inputs from different sources. Two main sources obviously which are of relevance here is input coming from outside and then the recurrent connection. Because in this description, you have only two kinds of inputs which are changing from one mode to another. The input coming from outside and then inputs that are coming from within itself, the recurrent connections. So the CA3 pyramidal neurons receive inputs from both from EC, that's input coming from outside. And these inputs, so CA3 itself has multiple layers in it. And so these layers are named as stratum lacunosum moleculae. Okay, some complicated Latin expression. Kind of sounds like some of these magic spells in Harry Potter, right? Then stratum radiatum and stratum pyramidal. So basically, this is the layer where you have the pyramidal cell bodies. Stratum is strata or layer. That pyramidal is pyramidal. Now, stratum radiatum is where you have radiation, that is connections of the pyramidal neurons among themselves. That is why it is called stratum radiatum. Stratum lacunosum molecular, hell, I don't know what it is, but anyway, that is the layer where the CA3 gets inputs from EC. So these are different layers, and now let us see what happens to ACH, or when ACH is applied to CA3. ACH suppresses the synapses in stratum radiatum, which is the recurrent connection. So this part, all right, a lot more than it suppresses synapses in stratum lacunosum molecular. So when you have ACH high, it suppresses the lateral connections, right? Which is what we wanted from a purely mathematical point of view. Okay, and now high ACH suppresses recurrent connections, therefore more than the inputs coming from this, and high ACH also increases LTP in ACH. So we all that also we have seen high ACH increases plasticity of the lateral connections. So both anatomy of CA3 and the kind of neurophysiology or the neurochemistry of ACH or its action on CA3 connections support the requirements, right, of CA3 playing the role of a memory storage system. So that is correct. Now we said, okay, for CA3 to work as a memory storage system, you need to operate in two modes. And there should be somebody who is operating between these two modes. So who is controlling this mode switch? Okay, we said ACH can change it. High ACH will put in one mode and in storage mode. Low ACH will put it in the retrieval mode. But who controls this acetylcholine? Who controls the modulator? Who modulates the modulator? So here we need to understand where ACH is coming from, right? And coming to the CA3, and how is it controlled by hippocampus? There must be some feedback from hippocampus to whichever part of the brain that is sending ACH to CA3. Let us see how that works out. Now, ACH, like I said, is a neuromodulator system, and neuromodulators are all the time released by these small neuronal clusters which are located deep in the brain, and they send projections out extensively to most parts of the brain, both cortical and subcortical targets. So if you look at cortical areas, one cortical patch sends projections on the cortical patch, and there is feedback. Now they have reciprocal connection. This is the kind of connectivity you see within cortex. But neuromodulators is a slightly different beast. These are small clusters of neurons, okay, and maybe a couple of lakhs of neurons. That's all. And these guys project extensively to the cortex, all over the brain, and also to some subcortical targets. So this is the nature of the neuromodulator systems. These are the kinds of neuronal pools that release neuromodulators. And because of their diffuse connectivity, these systems are also called diffuse projection system. Okay, so all the four neuromodulators are released by such diffuse projection systems. So when it comes to acetylcholine, there are two such networks, the nucleus basalis and medial septum. So nucleus basalis projects to cortical targets, releasing ACH to the cortical areas. Medial septum projects only to hippocampus. So this is of more relevance to us right now. So there's connections from hippocampus to medial septum, and then from septum back to hippocampus. And that is what is called the septo-hippocampal loop. Because obviously you need to have some such loop, because otherwise how will you decide when to store information and when to operate CA3 in storage mode and when to operate in retrieval mode, right? So something about the overall activity in hippocampus should tell the septum, the medial septum, that okay, now I must operate in storage mode, so send more ACH to CA3. Or now I have to operate in retrieval mode, send less ACH to CA3. So there is some kind of a communication between medial septum and hippocampus. So when this loop, septo-hippocampal loop is stimulated, it decreases neural activity in medial septum. So then the loop from medial septum to HC and backwards acts as a self-regulatory system for HC. And so the feedback from hippocampus to medial septum comes from CA1. Okay, and medial septum to forward connections, which is where ACH is released, right, from medial septum to hippocampus comes to CA3. So this one, and CA3 projects to CA1. So this is a loop. This is the loop that we're talking about. So Asselman colleagues propose that the above loop is capable of making sure that ACH is released just when it is required. Okay, so here the story goes like this, the theory goes like this. Now we imagine that all these states are stored in CA3. What are these states? These are states that of neural activity that come to internal cortex. And what comes to internal cortex? So we have this cortex here, the huge high dimensional state, which projects to EC, a low dimensional state. And the EC is now trying to store information. So this is CA1, so this is CA3, and so the idea is that the EC has the direct projections to CA3 and also has projections via dentate gyrus. So this information, this compressed form of the cortex, seems to be getting stored in CA3 by the inputs coming from EC to CA3. And so there's some information stored here, and if the and that is what is also sent to CA1. Now, what comes from CA3 to CA1 is a reconstruction, because CA1 retrieves the pattern and sends it to CA1, and what comes from EC to CA1 is a direct pattern. Now, the theory goes like this. So the CA1 compares the direct information coming from the EC, which is the original, and the reconstruction coming from CA3, which is the reconstruction, right? And compare these two. If both match, that means it infers that it's already a stored pattern. You don't have to do anything. So it's sends a signal to medial septum saying so it doesn't activate, and therefore it doesn't release ACH. Whereas if the original and deconstruction in CA3 do not match, because comparison is done in CA1, if they do not match, then it says sends signal to CA3 like in this loop, sends signal to medial septum like in this loop, right? And medial septum then releases ACH, and it says, hey, I don't understand what this pattern is. I've never seen it before, so you better store it. So it causes ACH release, and that leads to pattern storage. So this is a theory, but this is a theory. I used to love this theory, and I used to teach it. I've been teaching for many years, but I find a lot of holes in this theory. Right, I'll just talk a little bit about those holes, and we have been developing an alternative theory in my lab. I'll maybe talk about that later, maybe after a week or so. I'll talk about our lab's work from now on, right? And so just quickly summarize and discuss a little bit about the potential problems with this theory. So to summarize, we have said that hippocampus is a short-term memory store, which is well accepted. There's no problem with that. CA3 has some sort of off-wheel network. I think this is where the problem starts. This is where people have been talking about it since the times of David Marr, who is a big name in computer science and in vision and so on and so forth. But I think this is where the problem starts, because everybody kind of fell in line with that. Everybody started repeating the same thing over the last, I don't know, 30, 40 years. So then if CA3 is storage system, then there are two modes of operation, storage and retrieval. And ACH plays a role of it's a neuromodulator, and it controls the modes of operation of CA3 between storage and retrieval. Now the role of hippocampal network, right? We have talked about that and how this loop controls, right, the storage and the switching of the modes of CA1 between storage and retrieval. Now let us talk about some issues with this. One question is, it is saying what determines the storage or not non-storage of information in CA3 is this comparison, right? CA1 compares whatever comes directly from CA3, and whatever comes directly from internal cortex. What comes from CA3 is reconstruction. What comes from internal is original pattern. If the two match, it says there's no need for storage and doesn't tell CA3 to store it. If they don't match, it says the need for storage and tells CA3 to store it. But point is, just because some information is novel, that doesn't mean you will store it, right? I mean, okay, these days you are at home, so there is not much of information overload, I hope. But suppose you come back to campus and morning when you come to a class, right, you will see like hundreds of students all along the corridor. You may not be knowing most of them. And just because you don't know somebody, your brain doesn't struggle to memorize all that. What I'm saying is you don't try to memorize everything that you come across on the street just because it is novel, right? So brain must obviously have some notion of saliency. Is it important for me, right? Is it salient? So only then, so you need to apply, it needs to apply a further criterion of relevance or importance, right? And then decide whether to store it or not. Just because something is unstored yet, you don't go ahead and store it because that will overload your memory. That's a stupid way of operating your memory. That is right there. There is a big missing piece. Also, this theory doesn't talk about what happens after the short-term storage. Okay, you store everything, all this in CA3. After that, what? How much can you store in such a tiny structure? Okay, so how do you this long? Everybody knows that there is a transfer to long-term storage in the cortex, and a lot of that happens in your various sleep stages. Okay, so memory is a very exciting story involving deeply involving sleep states, right? For this transfer of information from hippocampus to cortex. But even that is not a complete puzzle. So how does that occur? This theory doesn't talk anything about that. But most important problem, I think the glaring error in this, which I too just casually accepted for a very long time, is this. Oh, okay, so we don't have that information here. So the estimates, let me try to pull it up from another slide. We still have some time. Okay, good. So the thing is, okay, like I just said, in the rat, there are 12,000 neurons. Sorry. Yeah, there are 12,000 neurons and the connectivity of about each neuron gets about, I think, what was it, 3,000 or 4,000 connections, right? Now, so obviously that's not a fully connected network. And such connections, that's a sparsely connected network, which is okay. And even such networks can have associative, can be used as associative memories. Only thing is the capacity for such networks will be different. The formula will be different. For Hopfield network, we have yesterday stated that the capacity is 0.14 times n. That's the capacity. Whereas when there's a sparsity, when the connectivity is sparse, the sparseness is given by the parameter A, which is 0.02, right, for this system. The capacity is calculated to be 36,000 patterns. Now, so which means that CA3 is capable of storing 36,000 patterns. So therefore, it's a great memory storage, and so we should all be excited about it. This is what people are saying. Now, how on earth will you be able to prove something like that? How will you demonstrate that? How will you demonstrate that in a RAT CA3, right, you can store 36,000 patterns? Has that experiment ever been done? Has this calculation ever been verified? It's not only not been verified, it's not even easy to verify, because so even if you train start some kind of a rat coaching center and train rats on lots of patterns, visual patterns or whatever, how will you demonstrate that a rat can learn 36,000 patterns, visual patterns or orders, right? Because rats are good at learning orders, smells, right? Or take anything, sounds. Can you design an experiment and demonstrate definitively that an animal can learn 36,000 patterns? This is the first thing. It's really moot point. Next thing, even if you can demonstrate that, where is the guarantee that all the memory is sitting in this tiny part of the brain called CA3 and not distributed all over the place? Okay, so two major drawbacks And this is, in fact, the CA3 idea, this whole CA3 story, or the story that CA3 is a recurrent network and therefore it's a memory storage system, rests on this idea that it has all these kind of low recurrent connections and you can store patterns, and people have even done computational this mathematical estimates about the storage capacity. This is the crux of the whole theory, right? This is the nub on which the whole theory revolves. And there is such a deep weakness because you cannot verify this. Okay, and so I will try to argue. I'll make a series of presentations over a few hours of discussion. I'll try to argue that there's a whole different way of looking at the hippocampal circuitry and understanding the hippocampus storage operations in a very different way. You don't need to look at CA3 as a storage system in that theory, right? And so another thing is, when it comes to this theory, there are only pockets of mathematical calculations, but most of it is just hand waving. It's just verbal theorizing. There is no computation model that is worked out completely with the entire hippocampus and where you show that this is how hippocampus is storing memories. So there's a little bit of math and lots of hand waving and verbalization. So I'll try to present our own theory about this maybe in a week or so. So I'll stop there.